Hi everyone. Now we are going to discuss a topic called microbial growth. The study about the microbial growth is most important for understanding the microbial physiology. In this part, we majorly discuss about bacterial growth. Firstly, what is a growth and how do we define a growth? Growth can be defined as an orderly increase of all chemical compounds in a cell or population. Yes, growth can be defined as an orderly increase of all chemical compounds in a cell or population. In multicellular organisms, an increase in the size of an individual is an indication of growth, isn't it? So if any plant or animal is growing in size, we will tell that they are having the growth. Whereas for unicellular organisms like bacteria, it leads to an increase in the number of individuals. That is, increase in the number of the cells is going to be referred as growth. So that's how the bacterial growth can be defined as an orderly increase in the cell size as well as increase in the number of cells. As the increase in the cell size we can't see with our naked eye, but obviously we are going to pay more attention on the increase in the number of cells, that is bacteria or microorganisms. Growth is an essential component of microbial functions because any microbial cell has only a finite lifespan, that is a limited lifespan. Bacteria mainly divide by binary fission. Uh, when it is going to have a binary fission, when a bacterial cell reaches certain size, it obviously divides to form the daughter cells. So here is a one bacterial cell. When it attains the maximum size, it is getting into divided into two, and this is going to be called as binary fission. One cell giving rise to two, and these individual cells again giving rise to two. That means altogether four cells, and these four cells again giving rise to two to each. So where it is becoming the how many? 4 plus 4, 8 cells. So that's how one cell, first 2 and 2 becomes a 4, 4 becomes a 8. So this is going to be called as a binary fission where one cell is equally getting divided into two cells. Now coming to generation time. What is a generation time? The time required for the cell to divide, the time required for the cell to divide, or the time required for the population to divide is called as a generation time. So that means this cell is going to give rise to the 2 and this time is going to be called as generation time. What is it? The time required for the cell to divide or the time required for the population to divide is called as generation time. Okay, now this generation time is going to be differed from in different type of microorganism. For example, take E. coli. It takes only 20 minutes of the time of getting one cell into two, but Mycobacterium tuberculosis will take of about 20 hours of time to get into the next generation, and the Mycobacterium leprae is going to take about 24 days to have this generation time. So that's how it is differing from microorganisms which are not having the same generation time. And this generation time is also going to depend upon the factors such as nutritional requirements, physical conditions like temperature, etc. Okay, under optimal conditions, this generation time determines the rate of growth of microbial cultures. So that's how there is a link between this generation time and the bacterial growth. So now we will discuss about this bacterial growth in detail in the form of a growth curve. So this the phases of a bacterial growth curve or the growth curve simply. So because of the bacterial small size, it is not convenient to study the growth and reproduction of the bacteria by taking an individual cell, isn't it? So if you take an individual cell, it's very difficult to study about its growth, reproduction, all the things. So what we should do for growth studies, bacteria are usually cultivated in a liquid medium uh, where its growth leads to an increase in the number of bacterial cells. That's why the definition of the bacterial growth is going to be defined as an increase in the number of the cells, which follows a definite course or pattern. When that bacterial count of such culture is determined periodically and plotted a graph or a curve on the graph paper, you are going to get a curve. and that curve is going to be defined as growth curve. And this growth curve is going to possess mainly four phases. What are those four phases? Lag phase, log phase, stationary phase, death or decline phase. So here, just remember, there will be a little bit confusion for many of the students, as I came to notice that lag and log, they'll always get confused and write the log as the first and lag as the second. So don't get confused. In the alphabets, A is the first letter when compared to O. So obviously in your this phase is also, lag phase is the first phase and log phase is the second phase, which is also called as exponential or logarithmic phase. So we will come in detail discussing about all these phases one by one now. So the first phase is obviously the lag phase. So here, when a bacteria are introduced or incubated into a fresh medium, increase in cell number will not occur immediately. That means no number increase will occur in the first phase. So what happens in this phase? So here what is happening is the bacteria is trying to adjust to the new environment and also tries to synthesize the new components such as cofactors, enzymes, ribosomes, which are required for its cell growth and cell division in the coming phases. Though no cell division is occurring during this phase, the cell remains physiologically active. That means it won't be in the dormant phase like without doing anything, isn't it? Next, the length of the lag phase depends upon the type of the bacterial species. That means how much time it have to try to adjust means depends upon the bacterial species, size, inoculum, then quality of culture medium, environmental factors such as temperature, carbon dioxide, all these things. Okay. Now, the average time taken by this lag phase will be 1 to 4 hours or sometimes 2 hours approximately, but even it may take off from 1 hour to several hours. So this is how the lag phase is completely just trying to adapt to the adopt the bacterial cells to the growth conditions that they are having. Okay, then coming to the second phase, that is a log phase. So up till now we have discussed about the lag phase, which is a smooth linear line parallel to the time. That means, and x-axis is going to have the time and the y-axis is going to have the number of bacteria. So if there is no increase in this line, means it is going to be the parallel. So as the time is going on, there is no increase in the number, so that's why it's a smooth linear line. And then coming to the second phase, that is log phase. So here you can see, so this log phase is also called as logarithmic or exponential phase. Here the number of cells increase in geometric progression. That means the 2 becomes 4, 4 becomes 8, 8 becomes 16. So like this there will be a rate at which they are going to increase. After every generation the cell number multiplies and becomes double. So this doubling of cells occurs at regular intervals. They are not going to do as they wish. They are going to occur at regular intervals with a constant growth rate. So that's why we are having a nice slope of the line in the figure. Okay. So during this phase, the growth occurs at maximum possible, alright, by utilizing all the nutrients that are present in the available environment. Cells are very small, obviously, because they are undergoing the constant dividing. So obviously they will be small in size. And the time taken for this lag phase will be of 8 hours. Okay. And the actual rate of the growth or the slope of the line in the figure depends upon the growth conditions. Though nutrients are present, if the environmental conditions are not uh, making them to divide, so obviously there will be lessen exponential, that is this log phase. The phase cannot continue indefinitely. That means going on like this, it will not occur. Why? Because the medium is soon depleted of nutrients and enriched with waste. That means their waste only, or accumulation of toxic products. So because of all these things, the log phase cannot continue indefinitely. So obviously they are going to again have a smooth linear line, which is going to be called as a stationary phase. But here in the stationary phase, what is happening? Let's see. After some time, the bacterial growth ceases almost completely due to the depleting of nutrients or accumulation of toxic products. There is a balance between the cell division and the cell death. So here only cell division is occurring, but in the stationary phase we're having the balance between the new cell arises and the old cell's death. So that's how there is a balance and we are having a smooth horizontal linear line or a curve. So there is no net increase or decrease in the cell number. Obviously, how many are arising, the same number is going to die. The duration may be from few hours to few days, and many bacterial species produce secondary metabolites in this phase. So that means the toxins, antibiotics, so all these things are going to be produced in this stationary phase. Not only that, mutations can also occur in the stationary phase only, and some spore forming bacteria produces the endospores also. Okay, and here if this type of the cultures are going to be stained at the stationary phase, they won't stain properly. That means the bacteria stain irregularly, where you cannot justify the staining result. And during this stationary phase, as I told you, you are going to have a very smooth horizontal linear part of the curve, and this stage of growth is also being called as cryptic growth. What we call it as cryptic growth. So this is all about the third phase called as stationary phase. So we have seen the lag phase, the initial adaptation stage, then this log phase, dividing phase, and the third one stationary phase. Yes, here also cell division occurs, but at the same time cell death also occurs because of accumulation of the secondary metabolites, that is the toxins or the wastage, all the things. Okay, and then it enters into the next phase that is going to be called as death phase, which is also called as decline growth. So this is a phase of growth curve in which the number of viable cells decreases or decline. If the incubation is continued after stationary, so this is our stationary, if still you are going to leave what is happening to study about them, the cells will enter into the next phase called as death or decline. The population reaches death phase due to the death of the cells, and this phase occurs due to depletion of nutrients or exhaustion of nutrients, and also accumulation of toxins. During this phase, a constant proportion of cell dies at regular intervals. So that's why you're going to have a graph like this. Okay, but it is going to be in the opposite direction. So the various stages of the growth curve are associated with both the morphological and physiological changes of cells. So this is how you are going to have your growth curve, and let's have a quick review of all the four phases once again. So on the x-axis, we are going to have the time taken in hours, and the number of the bacteria will be on the y-axis. So the first phase is the lag phase, where it is trying to adapt themselves to the growth conditions, that is the lag phase. And here in the second phase is the exponential or log phase or logarithmic phase, where you are going to have the continuous growth of the number of the cells, and sometimes theoretically it is going to be indefinite, but practically it is not going to be. And then coming to the stationary phase, where the cell death and the cell division both are going to have the equal number, so we are having a smooth linear horizontal line. And then coming to the death phase, where bacteria die faster than they multiply, and it is a negative log of your exponential phase, and this also occurs at a regular intervals. So this is all about the bacterial growth curve. Then what is the importance of or significance of bacterial growth curve? Why all are reading about this bacterial growth? So the study of bacterial growth curves is important when aiming to utilize or inoculate known number of bacterial isolate. For example, if you want to enhance plant growth by increasing the biodegradation of toxic organs, or production of antibiotics or neutral products, so obviously you are supposed to know how many bacteria are able to produce how much of the quantity of concerned organic compound. So at that, the bacterial growth studies is very important. Then the second one, bacterial growth kinetics and bacterial numbers in a culture medium, is also having a very prominent role both in the research field as well as in the commercial point of view. Now, the growth kinetics is also useful for assessing whether the particular strain of bacteria are adapted to metabolize certain substrates such as industrial waste or oil pollution, which is a major thing in our productions, isn't it? And not only that, in the biodegradation process also. Then the slope and the shape of the growth curves produced from bacteria grown with mixture of industrial waste products can inform scientists whether the bacteria can metabolize a particular, suppose if you are taking so many compounds and you want to test whether the bacteria is able to grow or not, then you can compare the bacterial growth of all this, the same bacteria with the different waste. So which is going to give the maximum growth curve, there you can find whether it is how much potential energy source it is using to grow. So that's how you can find the best industrial waste, how the bacteria is going to digest. So that's how these are all the significances of bacterial growth studies. Okay, so this is all. With this we are going to finish the bacterial growth curve. Okay, I hope so you understood well. It's also a very important topic for the first years of BSc microbiology. Thank you.